Class 8 students, and welcome to the biology class. At the end of this video, you will be able to indicate the role of T helper cells and define specific immune response. Our body is made from special cells and organs that are responsible to fight pathogens. These special cells and organs form the immune system, which is the system responsible to defend the body against any pathogen. We notice also that the first line of defense in our body is represented by the natural barriers like skin and mucus. Once pathogen crosses the first line of defense, the second line, which is the first line of immune response by which it activates white blood cells, intervenes. In this second line of defense, mean the first line of immune response, inflammatory reaction occurs. In this reaction, special types of white blood cells are activated, which they are macrophage and granulocyte. Also, phagocyte, it's a specific type of white blood cells that is responsible to make phagocytosis. Phagocytosis, it's the process by which phagocyte destroy all the different types of pathogens. Because you are saying different types of pathogens, so this immune response, it's non-specific because it recognizes and kills the pathogen regardless of its nature. But the question raised now, what happens after phagocytosis occurs? What if phagocytosis is not able to defend the body against pathogen? What do you think will happen? Will we die? Or a third line of defense is involved? Or there will be another kind of cells is alerted? In fact, after the failure of the second line of defense, our body needs help. It needs the intervention of a new white blood cells and a new line of defense, which is more specific and definitely more efficient. Let us now remember together the immune cells, mean the cells that intervenes in the immune system, which are the white blood cells. White blood cells consist of granulocytes, lymphocytes, and monocytes. Lymphocytes, it made from two types, B lymphocytes and T lymphocytes. Now, if we want to concentrate in the T lymphocytes, also it consists of two types, which they are T helper lymphocytes and T killer lymphocytes. In this video, we are going to talk about the T lymphocytes, and we are going to start by specifically T helper lymphocytes. How are the T helper cells are going to start working and be activated? This what we are going to see in the next part of this video. Let us now concentrate and understand the structure of T cells. As observing, the T helper cell express, show, on its membrane a protein molecules that we call it receptors. These receptors are not identical in all T helper cells, where each kind of T cells characterized by their own shape of receptors, and this is very clear in these two different kinds of T cells. Similar to T helper cells, the pathogens express on their membrane a marker molecules called antigens. These antigens represent the identity of each pathogen, and thus each pathogen have its specific antigen. Now, of course, you are asking yourself about the role of the receptors that found on the surface of T cells and the antigens on the membrane of pathogens. Concentrate please on the following slides to guess their role. In this video, we will see what is the role of T helper cell. Once pathogen enters the body, it will activate the immune response, by which the macrophage will allow the presence of the antigen fragments on their surface. So, macrophages with the antigen fragments are called antigen-presenting cell. Now the T helper cell is able to recognize this antigen and fit to it like the key and the lock. To understand more, please follow us in the following slide. Figure A represents an influenza virus holding spikes, which are a marker molecules on its membrane. So these protein spikes represent its antigen. In figure B, it represents a magnified figure of the antigens of the virus. Concentrate now please in figure B and now in figure C. We will notice the complementarity between the shape of the antigens and the receptors by which allow them binding together. So here we have the antigen of the virus and here the receptor of the T helper cell. Due to the complementarity between this antigen and the receptor, they will bind together and fit together like the key and the lock. Thus, each kind of T helper cell will recognize only one kind of pathogen based on its antigen. Observe please the following video in order to see how this binding occurs. So, once influenza enters the body, by its antigen, it will fit with its complementary receptor like the key and the lock. So, we can say that T helper lymphocytes identify pathogens and distinguish one kind of pathogens from another based on the antigens of these pathogens. After that, T helper cells are activated. So, we must have complementarity between the T cell receptor and the antigen of the pathogen in order to fit like the key and the lock. We state that in the previous immune response, which led by phagocytes, it's non-specific. What can we say now about the second immune response, mean the third line of defense? Will the T helper cell be able to recognize any antigen? Of course no. Thus, we say that this immune response will be specific, where each kind of T cells is able to recognize only one kind of antigen and thus will bind to it like the key and the lock. Example here, we have a T helper cell present on its membrane, its rectangular receptor, and in the pathogen present a rectangular antigen. If we observe clearly, there is complementarity between this receptor and this antigen. Thus, of course, they will fit together like the key and the lock. If we want now to define the specific immune response, simply we say it is a response directed specifically towards one kind of pathogen. As an application, we have this one kind of T helper cell that is present on its membrane a triangular receptor. And we have three different types of pathogens where each one expressed on its membrane a specific type of antigen. Pathogen A represents a triangular antigen, pathogen B represents a circular antigen, and pathogen C represents a rectangular antigen. Which pathogen do you think this T helper cell will bind to it? Observe them clearly, please. Great, it's pathogen A, where there is clearly complementarity between its antigen and the receptor of this specific T helper cell. In the second time, we are going to learn about the activation of T killer cell. Thank you for your attention and goodbye.